Hello, my name is Dr. Altura. I got my BS, MS, and PhD degrees from the UCLA School of Engineering and Applied Science. My areas of specialty, I've been involved in research for over 40 years, are electrochemistry, corrosion science, and material science. What I'm going to discuss today is a special application of Darwin's tree of life. Darwin, back in 1859, came out with his book, a famous book, and where he described the tree of life. And basically, what it says is that life, as we see it today, developed by total random processes, by chance. This is what we call evolution. And the way it developed is totally random, and it takes little steps over a long period of time. And that's the way all the millions of species that we find today on the Earth developed. Okay, now what we're going to do, we're going to simplify the tree, because this tree is very complicated. You're talking about all these million species at the top of the branches, and whatever happened in here with the fossils, etc., a lot of it's unknown. We're going to make it simple. Why? Because a lot of times I found out when I was a freshman at UCLA that when you want to solve a very complex problem, you can sometimes get an idea about the complex problem by simplifying it and looking at the simple model and then getting back to the complex problem. So we're going to do the following. I just want to adjust the camera for a moment here. There you go. Instead of looking at the whole tree, which is a question mark, we're going to look at just the single cell, which is the way Darwin started. Darwin says there was a single ancestor cell from which all life developed from. And we're going to look at just one branch of this tree, which is a human being. To further simplify things, we're not going to look at the whole human being, because the human being is made up of, believe it or not, 37 trillion cells, a number that I can't even imagine. So we're just going to look at one cell. We're going to look at one breast cell, for example. Secondly, we're not even going to look at a total human cell, because each human cell has about 20,000 different proteins which have different jobs. So we're just going to look at, basically, instead of a whole human being, instead of a human cell, we're going to look at just two proteins which exist in the human cell. These are very special proteins that what are called tumor suppressors, BRCA1 and BRCA2. Everybody's familiar with these nowadays. If these two proteins, or the genes that produce these two proteins are mutated, this will cause a large increase in the incidence of cancer, breast cancer, ovarian cancer, and even prostate cancer in males. So we're going to look at just these two proteins, and we're going to see how these got from the single cell to the human being. Now, when you have the genome is composed of genes. Each gene codes for a particular protein. In this particular case, we have, the way it works, DNA sends out there's a duplicate messenger RNA which is based on the code of the gene that we're trying to formulate the protein of. We send this messenger RNA to the ribosome, which is the factory which produces the proteins. The ribosome reads the mRNA and a protein is produced. The protein is basically a large chain of amino acids combined together. The average, I believe the average number of amino acids in the average human protein, I think, is between 300 to 400. Large. Now, but how do we get started with a single cell? What kind of single cell was it? Okay, well, we're going to assume it's a very simple single cell. That's what Darwin probably had in mind. It wasn't very complicated because the complications arose later. So how are we going to model this? Well, we're going to model it based on a very simple bacteria, which a lot of work has been done on with a project called the Minimum Genome Project. What they did is they took this bacteria called M. genitalium. It's an actual bacteria that causes actual human infections. A bacteria that normally has 482 coding genes for proteins. And what they did is they, one by one, knocked out these genes until they came down to a 382 proteins, and the genes that produce these proteins and code for them. And they found that after you go down below 382, the protein, the cell stops to function, it dies, it can't reproduce. So we're going to assume that that the single cell is somewhat like the M. genitalium cell with the 382 types of proteins. Now, if that's the case, we can make the following assumption beyond that. BRCA1 and BRCA2 are very complex proteins. This M. genitalium has very simple proteins, has no large complicated proteins. That's number one. Number two, the BRCA1 and BRCA2 are tumor suppressors. An individual cell does not need tumor suppression because an individual cell does not suffer from cancer. So basically, we're going to assume that this single cell, because of what I just said, does not have in its protein makeup either BRCA1 or BRCA2 because they're too complicated, and this single cell doesn't need tumor suppression. It doesn't suffer from cancer. So, when the protein is formed, as I said, it's a string of amino acids, that protein is totally useless. Why? Because it has to be folded in the correct geometrical configuration for it to work. If it's not folded up, it will not do anything. By the way, I want to mention one more thing. Darwin, in his time in 1859, knew nothing about the molecular complexity of life. That only came later with Crick and Watson, who defined the structure of DNA. So, but the proteins have a unique feature which I want to mention here. This is what's called the immobilizer. You know, when I was, I guess about 20 years ago, I bought a used car. I bought it from somebody, he only had one key. Bought the car, I drove for about a week, and then I wanted to make a duplicate key for my wife. So, I went, I think I was in a department store, I went to the key facility there, duplication facility. The guy ran my key and I had a duplicate. I gave the original key to my wife and I kept the duplicate. Well, the next morning I go out, I try to start the car, I go into the car opens up, the windows work, the radio works, everything works, but when I turn over the engine, turn the key, nothing, it doesn't start. So I run back in the house and I get my wife's original key. I put it in there and it works beautiful. It just turns over the first shot, it turns over. Didn't know what was going on. So I had a friend, I have a friend who's a locksmith, and I asked him, what's going on over here? I have two keys, they're identical. So he says, no, they're not identical. You have an immobilizer in your car. What does that mean? That means that the key, the original key you had, has a tiny integrated circuit in there, which produces a certain electronic code. And when you stick that key into the steering column, not only is there a geometric effect that the key has to fit into the key slot, but there's also electronic effect. Basically what it means is that the electronics has to be coordinated. If the code that the integrated circuit in the key is different from the code inside the steering column, the car will not start. And the same thing is true for proteins. They have, in order for them to work properly, they need both they have to be totally geometrically formed and also have an electronic component. when they interact with substrates or other proteins. If there's just one amino acid missing, this can create a situation where the protein becomes either partially or totally ineffective. So the code, the sequence of amino acids in this protein is very, very important. If there's a mutation which causes one of those amino acids to be different than what was supposed to be there, or the amino acid is missing, this is going to cause the protein to be either partially or totally ineffective. So now, let's go and look at these two proteins a little closer, the BRCA1 and BRCA2. First of all, what we notice, as we mentioned, the average protein in the human body has about 300 to 400 amino acids. These are very large. The BRCA1 has 863 amino acids in a chain, and BRCA2 is even larger, 3,418 amino acids. That's a very large protein. And what is the functions of these two proteins? It's tumor suppression. They prevent the cell from becoming cancerous, basically. How do they do that? They're multifunctional. It's only one thing that each one of these does in the cell. They have multifunctions, and a new function is discovered every few months that for either one of these proteins. For example, they are useful with other proteins in combination to detect mutations in the DNA. First of all, detection of mutations. Secondly, DNA repair. This is particularly important for BRCA2. BRCA2 is involved in double-strand DNA repair. DNA is two strands. Imagine a railroad track. Two rails. Imagine it's not on the ground, but it's up in the air. The BRCA complicated protein surrounds, it folds and surrounds the defective part of the double strand, this double rail, and then calls in worker proteins, other proteins, and coordinates those on the rail. And what happens is, it's an amazing process, the defective part of the double strand is removed, and a new double strand portion is put in to bring the protein back, to bring the DNA back to its original condition. It's also involved in other things. For example, cell cycle arrest. What is a cell cycle? The cell cycle is when the cell begins to divide and produce two other cells, cell division. The way this is done, first of all, in the cell cycle, the DNA has to be reproduced. There has to be complete DNA codes that are contained in the original cell, and then the cell divides into two. What these two proteins are involved in, if there is a malfunction in the DNA, these two proteins will arrest the cell cycle so the cell can't divide. Why not? Because you don't want a defective cell that can cause cancer to divide. And finally, they're involved in the apoptosis process. What is that? If after trying to repair all the defects in the DNA and they fail, it can't be done, the cell is programmed for death. In other words, the total cell is destroyed in order to save the human being. These are totally unique, which means they're not only different from each other, but they're different from every other protein in the human cell, except for BRCA has about a 5% ring. 5% of the BRCA1 has a ring component to it, which is somewhat similar to another protein. But BRCA is totally different. There's no other protein or no other gene which is similar to the gene of BRCA2. And let's take a look and see what happens when you have mutations, where there's a single point mutation in the gene for either one of these two proteins, or multiple mutations. What you get is the following. For BRCA1, they've already found over 1,700 different mutations. And 850 of these are clinically significant. In other words, they cause cancer, because the BRCA1 is not doing its job as a tumor suppressor. And one more thing, these two proteins and the genes that code for these proteins have a tremendous amount of information, which the single cell does not have. Now, one more thing which is most important, Goldilocks. Again, we get back to the immobilizer. There's only one combination, one correct combination, which will make the immobilizer work properly and start that engine. The same thing for these two proteins. It has to be just right. If there's one amino acid out of place, or one extra amino acid in there, then you don't have the right combination. It's not just right. So there's only one combination which works. Let's take a look and see how many combinations we have for both of these for both these proteins. First of all, let's take a look at a simple example of a necklace. One around the neck. Let's say we have 5 types of pearls. Here I just show 4. Square, a triangle, a circle, maybe a hexagon, maybe one more, maybe an octagon. There's 5 different kinds of pearls, but the necklace only has 4 pearls, 4 positions. So how many different combinations are there? Well, you take the first pearl, 5 different combinations, multiply by the second pearl, 5 combinations, multiply by the 5 in the third pearl, 5 in the fourth pearl, you get 625 different combinations for 5 types of pearls and 4 positions. The general formula for the number of combinations is the number of types to the exponent p, which is the number of positions. For example, there's 5 types to the 4th power in this particular example. Now, amino acids, which again, all proteins are formed from amino acids. How many amino acids are there? How many types of amino acids are there? There are 20 different amino acids for life. Actually, originally in the physical universe, in the physical world, we had 40, because each amino acid has a mirror image. Now these look similar, but they're not identical. But for some reason, which we don't know why, in life, only the left-handed amino acids are used. We don't know when a single cell came about whether that was the case at that point. So we could have as many as 40 different types, but we're going to stick to 20. So using this formula, how many different combinations do we have for BRCA1? Well, we have 20 different types, and the number of positions is 1,863, so it's 20 to the 1,863 power. BRCA2 is even bigger. The combinations are 20 to the 3418 power. It's a tremendously large number. 3,418 zeros. If I was to pronounce a zero every half a second, it would take me over a half hour just to pronounce all the zeros in the BRCA different combinations. But to get an idea of how large these numbers are, and again remember, there's only one right combination that will work for both of these, for either one of these. Let's take a look at Powerball. The total number of combinations of Powerball is somewhere around 1 billion, 10 to the 9th power. 9th power compared to these two powers here, it's minuscule. And still Powerball is very hard to win. How many people win at Powerball? So let's take a look at something much larger than that. Let's take a look at the whole observable universe. How many, the observable universe, based on what we know now, is composed of trillions of galaxies. And each galaxy is composed of many billions of stars. The number of stars, for example, in our own Milky Way galaxy is about 100 billion. But the Andromeda galaxy, which is our neighbor, has over 350 billion galaxies, 350 billion stars. And then we're talking about trillions of galaxies, and we're talking about a universe that's billions of light years wide. We're talking about a very, very large number of atoms. They figured out how many atoms there are in this observable universe. 10 to the 80. Even though we're talking about such a large universe with so many galaxies, so many stars in each galaxy, and each star being composed of so many trillions and trillions of atoms. The number of atoms, the total number of atoms in the universe is only 10 to the 80 compared to these two large numbers. And if you take the number of particles in the universe, you separate the atoms into protons, neutrons, electrons, neutrinos, whatever, you only get 10 to the 86 compared to these large numbers. Can you imagine looking for one neutron? Somehow this neutron is special. It has only one combination that you want in this neutron. Flying around the whole universe at the speed of light trying to find this one neutron. And that's nothing to try to find this one combination for each one of these two. It's minuscule. Look at look at these numbers compared to these numbers over here. You can't even compare the number of atoms in the universe compared to these two large number of combinations for both BRCA1 and BRCA2. Let's take a look at one more thing. I call it the path is not important. What does that mean? That means that, you know, in Powerball, where they have two machines, one machine with 69 balls and one machine with 26 balls, the white balls are 69, red balls are 26. They pick 5 white balls out of there and 1 red ball. When they do this, they actually have more than one machine. They have at least 4 machines of each kind, 4 white machines and 4 red machines. And they pick those right before they're going to do the Powerball lottery at random, so that no one can mess around with these before. So we have 4, 1 out of the white ones and 1 out of the red ones. Now, no matter which Powerball machine you pick, the odds will still be the same. If Joe Smith ends up winning the Powerball with that one number, the odds for him are going to be the same whether they use one of those machines versus another machine. Same thing for the red machine. And let's assume you think that somehow Powerball is biased because of the way the air blows up. It's not totally random. So let's take a PC. We can generate random numbers on a PC also. And these are very random. They've determined this. So let's assume that on the PC that we have random, we pick out random numbers. The same thing, 6 numbers out of 69, I'm sorry, 5 numbers out of 69 numbers and 1 number out of 26 numbers. Well, let's say you're afraid that the computer was hacked or something. It's again not totally random. So let's take something that was around 200 years ago, a roulette wheel. So we have 2 roulette wheels, a white one with 69 numbers and a red one with 26 numbers. We spin the white roulette wheel 5 times, and we spin the white we spin the white roulette wheel 5 times, and we spin the red roulette wheel 1 time. The odds are still the same, whether we go with the 4 different Powerball machines, whether we go with the PC, and whether we go with the roulette, which is 200 years old. So the same thing applies over here. These odds, which are so much greater than the odds of finding a single atom in the universe, a single particle in the universe, apply to this single cell here. The single cell, I'm talking about Darwin's tree, the single cell did not have these 2 proteins. They're complicated. Single cell doesn't need them because it doesn't have cancer. And we know that the human cell has these 2 proteins. Whichever route the evolution took to get from the single cell, which doesn't have these 2 proteins, to the human cell, which has both of these, the odds are the same. The odds are still this high What do these odds mean? That means, you know, for this to work, you have to have cells generating cells. You have to have a lot of generations of single cells going to multiple cells. But the number of generations you would need here, even in an ideal case, where you assume that every time that a cell divides, it produces a mutation, which is not true, because usually mutations are only produced 1 in a 10,000 or 1 in a million, depending if it's a human being or a single cell. Also assume that all the mutations are good mutations. In real life, only 1 out of 500 or 1 out of 1,000 mutations are good. The other ones are bad or neutral. But assuming all this, you still need billions and billions and billions of generations to get that one single combination for either one of these. And you need billions times billions times billions of years to be able to do this. And we know that our Earth or our universe is not that old. So this even applies to the average protein in the human cell. The average protein in the human cell, well, let's go a step lower. Let's take the average protein in a bacterial cell. I think the average protein is somewhere around the average protein in a bacterial cell is somewhere around 250 to 300 amino acids. So let's say you have 250 amino acids for this original cell here, average protein. Well, 20 to the 250th power is still so much larger than 10 to the 80 power. They're not comparable. And here, to cut to a human being, not only did you have to produce BRCA1, BRCA2, and you have to produce 20,000 different proteins. You have to have 20,000 different genes that have to be incorporated to produce all these proteins. Each one would take more than the age of the whole universe. Let's take one final example. Titin, the largest protein in the human body. Titin is the springiness in muscle tissue. Is formed by titin. Titin does not have 3,400 amino acids in the chain. It has 34,000 amino acids. And they all have to be in the right order, the right combination. And this protein has to be folded properly. If anything goes wrong and the amino acids are not in the right combination, one is missing or whatever, this can cause muscular type diseases. You can look up in the literature. One more final note before I leave. We're not talking here about some theoretical argument between evolution and intelligent design. We're not. Because we're talking about something very practical here. We're talking about saving many thousands of human lives to be able to somehow get these two proteins back into functioning situations so they can act as the original tumor suppressors. This is no joke. This is not simply a theoretical argument. And I know in my 45 years of research that I've done, perspective, how you look at a problem, is very, very important. Let's make sure that our perspective here is correct, because we're talking about saving the lives of thousands and thousands of people. Thank you very much.